Welcome to Let's Learn Plants. I'm absolutely thrilled to be your guide for today's tutorial explainer. We're going to dive deep into the microscopic, complex biochemical pathways of photosynthesis. Seriously, we're looking at how plants pull off one of the most miraculous feats on Earth, taking invisible carbon dioxide right out of the air and transforming it into the literal physical building blocks of life. It's like magic, but it's chemistry. Here's our syllabus for today's lesson. We'll start with an overview of carbon reactions, move into the C3 Calvin-Benson cycle, and then look at the C2 Photorespiratory flaw. After that, we'll cover the C4 cycle upgrade, CAM, the desert adaptation, and finally, wrap up by comparing these carbon fixation pathways.
Part 1. Overview of carbon reactions, the foundation.
Part 2. The C3 Calvin-Benson cycle, the primary pathway.
Okay, let's jump right in. The carbon reactions, which you might have heard called the dark reactions in the past, actually happen in the stroma of the chloroplast. Their whole job? To reduce atmospheric carbon dioxide into carbohydrates. To pull this off, they rely entirely on the energy stored in ATP and NAD-PH. You know, those high-energy products we get from the light-driven reactions? The absolute most universal pathway for this autotrophic CO2 fixation is the Calvin-Benson cycle, and it operates in 3 highly regulated phases: carboxylation, reduction, and regeneration. Let's look at phase 1, carboxylation. This phase stars what is arguably the most important enzyme on the entire planet, ribulose 1,5-bisphosphate carboxylase oxygenase. We all just call it Rubisco. So, Rubisco takes 1 molecule of CO2, 1 molecule of water, and covalently bonds them to a 5-carbon skeleton called RUBP. Boom! This reaction instantly gives us 2 molecules of 3-phosphoglycerate, or 3-PGA. And hey, since 3-PGA is a 3-carbon molecule, and it's the very first stable intermediate formed here, we call this the C3 cycle.
Moving into the second phase, reduction. This is where we actually spend that stored energy. First up, the enzyme 3-phosphoglycerate kinase steps in. It uses ATP to phosphorylate our 3-PGA, turning it into 1,3-bisphosphoglycerate. Immediately after that, NADP-glyceraldehyde 3-phosphate dehydrogenase uses NADPH to reduce it, creating glyceraldehyde 3-phosphate, or G3P. Here's the math. For every 3 CO2 molecules that enter, we get 6 of these G3P molecules. 1 leaves the cycle to become plant food, like starch or sucrose, but the other 5, they have to stay behind. So why do those 5 G3P molecules have to stay? Well, that brings us to phase 3. For this whole process to actually be a cycle, the plant absolutely has to regenerate its initial carbon acceptor, RUBP. This doesn't just happen magically. It requires this intense, tightly choreographed 10-step enzymatic cascade to reshape those 5 3-carbon molecules back into 3 5-carbon molecules. Let's meticulously track these carbon skeletons through the first 5 steps. Step 1. Triose phosphate isomerase converts a G3P into DHAP. Next, aldolase merges a G3P and a DHP to create fructose 1,6-bisphosphate. Following that, fructose 1,6-bisphosphatase strips away a phosphate to make fructose 6-phosphate. Got it? Okay, step 4 brings in trans-ketotolase to react G3P with that F6P, producing a 4-carbon sugar, erythrose 4-phosphate, and a 5-carbon sugar, xylulose 5-phosphate. Finally, aldolase strikes again, combining erythrose 4-phosphate with another DHAP to form the 7-carbon cetoheptalose 1,7-bisphosphate. It's like a giant biochemical puzzle. Hang in there, class. We are almost at the finish line for regeneration. Step 6 uses cetoheptalose 1,7-bisphosphatase to dephosphorylate our 7-carbon sugar into cetoheptalose 7-phosphate. Transketolase then reacts it with our last G3P to make ribose 5-phosphate and another xylulose 5-phosphate. Now, phosphopentose epimerase and phosphopentose isomerase act like little structural sculptors, converting both of those molecules into ribulose 5-phosphate. Finally, phosphoribulokinase spends 3 molecules of ATP to successfully regenerate ribulose 1,5-bisphosphate. And just like that, the loop is closed.
Part 3. The C2 Photorespiratory flaw, a biological problem.
But, nature is rarely perfect. And this brings us to a fascinating evolutionary biological problem called the C2 oxidative photosynthetic carbon cycle. Honestly, we usually just call it photorespiration. It's essentially a biochemical flaw that plants have to constantly manage. The whole issue comes down to comparing Rubisco's productive side with its, well, wasteful side. We know Rubisco binds CO2. That's its carboxylase activity. But unfortunately, it can also bind oxygen. When it accidentally grabs oxygen instead of CO2, it produces one useful 3-PGA, but it also creates a toxic molecule called 2-phosphoglycolate. This byproduct literally inhibits crucial chloroplast enzymes. So to survive, the plant has to clean up this mess using the C2 cycle.
This cleanup effort is an incredibly complex salvage pathway, spanning 3 entirely different cellular organelles. It starts in the chloroplast, where 2-phosphoglycolate phosphatase neutralizes the toxin into glycolate. The glycolate is then shipped over to the peroxisome. Once there, glycolate oxidase oxidizes it into glyoxylate. But that produces dangerous hydrogen peroxide as a byproduct. Yikes. Thankfully, peroxisomal catalase immediately shatters that hydrogen peroxide into safe water and oxygen. After that, glutamate glyoxylate aminotransferase converts the glyoxylate into the amino acid glycine.
Now let's move over to the mitochondria and see how this builds into an actual energy drain. The glycine enters the mitochondria, where an enzyme called glycine decarboxylase takes 2 glycine molecules and oxidatively decarboxylates them. This reaction releases NADH, ammonia, and, crucially, a molecule of CO2. That CO2 represents carbon that the plant had previously spent hard-earned energy to capture. So losing it, it's a terribly wasteful loss. Finally, serine hydroxymethyltransferase merges the remaining components to form the amino acid serine. Okay, we have to trace this carbon back to safety. The newly minted serine travels all the way back to the peroxisome. There, serine 2-oxoglutarate aminotransferase converts it into hydroxypyruvate. Then, NADH-dependent reductase changes that into glycerate. Finally, the glycerate re-enters the chloroplast. The enzyme glycerate kinase uses ATP to phosphorylate it, successfully regenerating 3-phosphoglycerate, which can finally drop right back into the Calvin cycle. The plants survived the error, sure, but man, it cost a whole lot of energy.
Part 4. The C4 cycle upgrade. Overcoming limitations.
So what exactly happens when plants live in super hot environments where this wasteful photorespiration would be downright fatal? Well, they evolve. Let's introduce the C4 carbon cycle. This is a brilliant metabolic upgrade used by plants like corn, sorghum, and sugarcane. It completely overcomes the limitations of the C3 cycle, especially in hot, dry climates. The secret to the C4 upgrade is a physical architectural marvel called Kranz anatomy. C4 plants possess this specialized leaf structure where a tight ring of bundle sheath cells is wrapped by an outer layer of mesophyll cells. This creates a distinct spatial separation. The plant purposefully isolates Rubisco deep inside the bundle sheath cells, far, far away from atmospheric oxygen. It effectively creates a super powerful carbon concentrating mechanism. Here's the brilliant chemistry behind it. Out in the mesophyll cells, an enzyme called PEP-CK, which by the way has 0 affinity for oxygen, grabs CO2 and attaches it to PEP, forming a 4-carbon acid called oxaloacetate. This is then converted to malate or aspartate and physically smuggled into the inner bundle sheath cells. Once it's safely inside, it gets decarboxylated. That means it drops off the CO2 directly onto Rubisco, basically flooding it with carbon and totally preventing photorespiration. The leftover pyruvate then goes back to the mesophyll, where pyruvate phosphate kinase spends 2 ATP to regenerate the PEP. Yes, it costs more energy up front, but the efficiency gained is absolutely massive.
Part 5. CAM, the desert adaptation.
If C4 is an upgrade for hot climates, what about the most extreme, dry environments on Earth? Enter the ultimate desert adaptation, Crassulacean Acid Metabolism, or CAM. This is the survival pathway chosen by succulents, cacti, pineapples, and orchids to thrive where water is incredibly scarce. While C4 plants uses spatial separation, physically doing the different steps in different cells, CAM plants use a temporal separation. A CAM plant performs its initial CO2 fixation at night and then runs the rest of the cycle during the day, all within the exact same cells.
Let's look at the daily biochemistry of a CAM plant. At night, when it's nice and cool, the plant opens its stomata. PEP-CK captures CO2 to form oxaloacetate, and then an enzyme called NADP-malate dehydrogenase converts it to malate. The plant literally pumps this malic acid into massive cellular vacuoles to store it overnight. Then, during the blazing hot day, the plant seals its stomata tightly shut to prevent any water loss. It pulls that malate out of the vacuole, decarboxylates it, and releases the hoarded CO2 to Rubisco, so it can run the Calvin cycle completely safely behind closed doors.
Part 6. Comparing carbon fixation pathways.
We have covered some truly incredible ground today, traversing the complex biochemical landscapes of plant life. Now, let's bring it all together. Looking at the synthesis, we see the C3 cycle is our baseline. Used by most plants, but it suffers from high photorespiration and relies purely on daytime fixation in the mesophyll, yielding a 3-carbon product. The C4 cycle, seen in maize and sugarcane, utilizes Kranz anatomy for spatial separation. It yields a 4-carbon product, basically minimize photorespiration. Finally, the CAM cycle, used by cacti, uses temporal separation. It fixes carbon at night to produce malic acid, perfectly balancing extreme water efficiency with minimized photorespiration.
As we wrap up this explainer, I want to leave you with a macro-level ecological thought. We've just seen how C4 and CAM plants evolved specifically to counter the flaws of Rubisco under high heat and low water. So, as global temperatures continue to rise, altering the solubility of atmospheric CO2 and increasing heat stress, which of these intricate metabolic pathways do you think will dictate the future survival of our planet's vegetation? Keep thinking about that, keep questioning, and until next time, class, keep learning.